Hello everyone, this is Ankita Tripathi, working as an associate professor in Amity College of Pharmacy. Today I'm going to discuss on a topic blood formation. Before proceeding, what exactly blood formation is, how it is occur, what is the mechanistic behind the occurrence of blood. So let's have a quick view on what is blood. Blood is defined by different authors in a different way. What they have a common in is that it's a fluid connective tissue. Fluid means it is highly viscous in nature. That makes it different from water. Connective tissue as it travels all along the body, so it is connecting one organ to another, entire body to entire part. So we can just say that blood is a fluid connective tissue which take away all the metabolic waste from the cells and deliver necessary new substance like minerals, iron, oxygen to them. So we can say that if a life exists in the cell, it helps. It is done by the blood. Blood is further divided into two parts. One is a solid part, and another is a liquid part. Let's have a view on the composition of a blood. If we go on this diagram, we will see that total body weight is divided into two categories. One is the other fluid and tissue, which is covers 92% of the body, and 8% is constituted by our blood. Blood is further divided into plasma and formed element. If we consider 8% is 100% of a blood, in that 55% is a plasma. Plasma is a liquid part of the blood. It means the fluidity and the connection is provided by them. And 45% is a formed element, which constitute RBC, WBC, and platelets. Let's have a look on plasma part. Plasma part is a liquid part which maintain the fluidity and connectivity between the cells, as it is yellow in color when it is separated by the RBC, because RBC have a hemoglobin in it, and due to hemoglobin the color is red, and when we separate or centrifuge plasma from the formed element, it turns to yellow. So we can say that plasma have yellow in color, or plasma have a yellow color. The plasma is further divided into three parts. First is protein, which constitutes 7% of it. Another is water, which constitutes 91% of it. And third are miscellaneous or other solid, which constitute 22% of it. In protein part, we have four fundamental proteins in it. What is albumin? Albumin is also known as transport protein, which because it helps in the transportation of mineral nutrients to one part of the body. Other is globulin, which is also known as immune protein. It helps in the formation of antibody. So we can say that it helps in our defense mechanism. Third and fourth are the clotting factor, first and second. They help in the coagulation and activation of platelets. So we can see that if we have to stop bleeding or we have to prevent hemorrhage, both these clotting factors are required, because at last, at the third step, fibrinogen get converted into unstable to stable fibrin. This fibrin is known as clot, and it is formed because of the presence of fibrinogen. The 91% is water. So we can say that the fluidity and the traveling or transportation is easily done because of the water part. Other 2% is a solute, which constitute ions, which provide essential energies, nutrients, which provides us balancing for waste products such as urea, kerosene, and uric acid, gases like oxygen, nitrogen, and carbon dioxide, and laboratory substances like hormones, amines, etc. Now, this was the part for the plasma. Now, let's move to formed element, which is constitute 45% of 100% of your blood. Formed element is further divided into three parts. One is RBC, which is a solid part, WBC, which is also solid part, and platelets. RBC is also known as erythrocytes. Generally, erythro is a word derived from a Latin word, which means erythrites. It is because of the red in color. Erythrocytes have a lifespan of 7 days and it consists of iron-rich pigment known as hemoglobin. Due to presence of hemoglobin, our RBC is red, and this RBC red color helps the entire blood color to be red. Now, coming to leukocytes. Leukocytes, generally known as WBC, that is white blood cells. These are colorless substances because they don't have hemoglobin in it. So we can say that WBC are colorless. Further, WBC is divided into two parts. One is granulocytes, another is agranulocyte. This division depends on the presence and absence of a granule. Granulocytes means presence of a granule. Agranulocytes, A means absence. So absence of a granules is known as agranulocytes. Granulocytes consist of three parts. One is eosinophils, other is basophils, and third one is neutrophils. Eosinophils is in substances which is dyed with an acidic color. It constitutes of 2 to 4% of your WBC. If I consider 100% of WBC in which we have only 2 to 4% of eosinophils, it protects us from allergy and asthma. It is generally formed in the bone marrow. Now coming to basophil, basophil was the name given by the staining of a basic dye. That is why it is known as basophils. It constitutes about 0.5 to 1% of entire WBC. It contains a larger cytoplasmic granules, which plays an important role in immune responses. It also protects us from the allergic reaction by releasing histamine and other inflammatory mediators. It also helps in secretion of histamine, serotonin, and heparin. It have a vital role in asthma because it causes vasoconstriction. Now coming to the third part, which is a neutrophil, it constitutes about 60 to 70% of entire WBC. Neutrophil is generally found in the bloodstream and also present in the pus. It constitutes about 60 to 65% of WBC. It has 2 to 5 lobe nucleus. It is generally stained with neutral dye. That is why it is known as neutrophils. Apart from that, it is the first immune system to respond against the invaders present from outside the body. The lifespan of a neutrophils is up to 8 hours. After that, it dead, and they are generally produced every day in the bone marrow. Now we have agranulocytes with us, that is monocytes and lymphocytes. The name monocytes is only because of the presence of a single kidney-shaped lobed nucleus. That is why it is known as monocytes. It constitutes about 3 to 8% of your WBC. It is also known as garbage trunks because it carries all the pathogens, debris out from the body. Lymphocytes are generally formed by lymphoid tissue. That is why they are known as lymphocytes. They are generally divided into 3 parts: B cell lymphocyte, T cell lymphocyte, and natural killing lymphocyte. These are the first who kills or digest the solid pathogens by the process of phagocytosis. Now coming to the exact topic, what a blood formation is. Blood formation is generally known as hemopoiesis or hematopoiesis. It occurs in the bone marrow. Bone marrow is present in our long bone. For example, humerus, femur, flat bones like rib and cranial bones. How does it occur? When the sperm and the oocytes mate together, fertilize together, they form a fertilized egg. After these fertilized eggs reaches to Marulla stage. Marulla stage is prior to Vesicular stage, where these eggs stay for 3 to 8 days and they get converted into 8-cell embryo. These 8-cell embryo are further after differentiation and the nucleation, it gets turned into Vesicular phase. In Vesicular phase, they are double-sided and single-sided cells, and it is the 31st stage of the nucleation. After that, the nucleus is ejaculated from the cells and it's converted into human embryonic stem cell or pluripotent cell. Then this human embryonic stem cell or pluripotent cell is then converted into 3 cells, that is ectoderm, mesoderm, and endoderm. The mesoderm part of the pluripotent cell further differentiate into the circular and lymphatic system, muscle system, skeleton system, kidney, reproductive, and the dermis layer of the skin, because only the dermis layer have the blood vessels. Epidermis and mesodermis, they don't have the blood vessels with it. The endoderm part just form the inner lining for our organs like pancreas, digestive tract, thymus, etc. Ectoderm form the epidermis, that is a covering layer which provides the cushions to our body, or we can say that first layer of our epidermis is a corneocyte because that is a dead body, a dead part. It helps with the production from the foreign particles traveling inside the body. Other than it helps in the formation of nerve cells, resulting in nervous system and sensory organs. As the formation of WBC, just have a quick recall it. As the formation of blood have 3 main different stages. First, formation of RBC. Second, formation of WBC. Third, formation of platelets. As what I have discussed earlier, that when the sperm and the oocyte combine, they turn into fertilized egg. This fertilized egg just reaches to Vesicular stage after reaching the Marulla stage and converted into pluripotent cell. This pluripotent cell is further differentiated in 2 cells. One is your myeloid cells and another is lymphoid cells. Myeloid cell is a precursor for RBC formation, WBC formation other than a lymphoid, and platelets formation. So let's have a quick review what is and how erythropoiesis occur. When there is a condition of hypoxia, it means that when the level of oxygen is decreased in the body, the kidney releases a hormone known as erythropoietin. When this erythropoietin reaches to the bloodstream, it reaches to our pluripotent cells and it differentiates myeloid cells. Myeloid cell then is further converted into your pro-erythroblast because all the process of nucleation occur in the blastula phase. Then the pro-erythroblast. Then pro-erythroblast then further undergo nutrition and differentiation and converted into erythroblast. Then this erythroblast is converted into pro-erythrocytes and further it is formed as an erythrocytes. And this erythrocyte then reaches to our bloodstream and perform the function. Same time, the mitochondria of our body also form a pigment which is known as hemoglobin. Hemoglobin have a hematinic acid with it. So because of the presence of hematinic acid, our blood turns to red. So the entire system because of RBC and hemoglobin is red. Now coming to the second part of blood formation, that is leukophoresis. Leukophoresis is a main concern because it has been divided into two parts, granulocytes and agranulocytes. A lymphopoiesis or lymphopoiesis is occurred due to the presence of colony stimulizing factor, that is CSF. What happens is also formed by the precursor myeloid stem cell. Pluripotent cells get converted into two cells, one is myeloid and another is lymphoid. When on the myeloid cells, when our CSF reaches, it converts our myeloid cells into myelocyte cells because myelocytes are the precursor for the leukocytes. Then the myelocytes is converted to promyeloblast, then to myeloblast, then myeloblast is converted into promyelocyte, and then further it's converted into myelocyte. This myelocyte is actually what a WBC is. Now, the WBC is further divided into two categories, as we all know. One is your agranulocytes, another one is your granulocyte. The granulocytes moiety is differentiated only because of the presence of granules, and these granules are generally a dye which impart a color to it. Then our myelocytes is further converted into eosinophils, which forms eosinophils. As we all know that eosinophils is protect us from allergy and asthma. It constitutes 2 to 3% of WBC. It combats infections. It is highly present in the in digestive system. Basophil is formed by the conversion of myelocyte into pro-basomyelocytes to basocyte. This basocyte is known as your basophil. Basophil was generally named by the dyes that is known as your basic dyes, which is staying basic in color, and it have a very high I can say it is a very high responsibility and vital role in the immune system, and it helps in the it has a vital role in the asthma because it's called bronchoconstriction. It protects us from allergic reaction by releasing histamine and also by dilating our vessels. It also secretes serotonin, histamine, and heparin. And last part of granulocytes is neutrophils. Neutrophils is named after the dye names as neutral dyes. What happens when myelocytes get converted into neutrocytes, then it's to neutrocyte. This neutrocyte is known as your neutrophil. What happened? Neutrophils are basically neutral dyes, which constitute about 60 to 65% of WBC. It is also present in the pus, and it acts as a strong it works as a strong oxidant because it helps in the destruction of bacteria. And then coming to the agranulocyte part of the WBC. WBC is further converted into agranulocytes. In agranulocyte, we have two parts. One is monocytes, which is also formed by the myeloid cells. Another is your lymphocyte, which is formed by the lymphoid tissues. Because a pluripotent cell is converted into two parts, myeloid cell and lymphoid cell. So lymphoid tissue helps in the production of your lymphocyte. When B lymphocytes are formed, when B cells are formed, when T lymphocytes are present, then T cells and the NK cells are formed. Now, the last part of our topic, our session, is your thrombopoiesis. Thrombopoiesis generally means the formation of platelet or thrombocyte. It also happens by the process of pluripotent differentiation into myelostem cell. What happened? when the thrombopoietin, which is present in the kidney and the liver, reaches to the myeloid stem cell is get converted into megakaryocytes. This blast then is converted into your megakaryocytes because it have a very abundant or very high numbers of nucleus in it. Then this is converted into your platelets. Platelets generally help in the process of coagulation. As platelets activate themselves when the blood reaches to the when blood atmospheres to the oxygen, it converts this to hematinic, and this help in activation and regulation of platelets. And after the activation and aggregation of our platelets, our clotting starts. So they said that if platelets will not activate, no clotting will happen. So platelets have only one importance, that will help in the coagulation. This was for all the today. Thank you and have a good day.